Hello, everyone. I'm Linda Tintawi, CEO of the LustGarten Foundation, and welcome to Progress is Paramount, our annual review of the accomplishments we've made and how this knowledge is forging an exciting path forward in 2025, a year we believe will mark a pivotal turning point in our mission to accelerate and expand life-saving treatment options for patients. Today, you will hear from the three leaders responsible for guiding and driving this progress. LustGarten's Vice President of Research, Andrew Rakeman, our Chief Scientist, David Tuvison, and our Chief Medical Advisor, Elizabeth Jaffe. Our research would not be possible without all of you. Thank you. You have my commitment and the promise of our researchers in the United States and Canada that our shared path forward will not waver. Progress is paramount, and we will continue to work together to transform pancreatic cancer into a curable disease. We encourage you to ask your questions by typing them in the Q&A box below. And it's now my great pleasure to welcome Dr. Andrew Rakeman, LustGarten's Vice President of Research. Andy, welcome. We have so much good news to cover today. Looking forward to the great conversation. And Dr. Rakeman, take it away.
Thanks, Linda. And I agree, there's a lot of great stuff to talk about today. And I have two of the best people to talk about it with, as you mentioned, our Chief Scientist, Dr. David Tuvison, and our Chief Medical Advisor, Liz Jaffe, who not only serve in important leadership roles at their respective institutions, Cold Spring Harbor and Johns Hopkins, but where they each lead a LustgartenLIVE program there and work with us at the foundation, as you say, to drive progress to set our agenda moving forward. So Dave, Liz, welcome. Thank you for joining us here today and to take a look back on all of the wonderful progress that's happened in 2024 and how that's shaping our outlook for 2025 and beyond. So we have Dave and Liz popping up on the screen now. Welcome and thank you for being with us this afternoon. So maybe before we get into some of the specifics that we're going to talk about today, and I know we have a few important highlights that we want to touch on, but I thought I'd give you each a chance to start off and kind of give a perspective on why you think right now is an exciting time for pancreatic cancer research and what's really giving us some momentum and giving us some hope for the future. So maybe, Dave, I'll give you the floor first. If you can give us a kind of quick intro to what we're going to hear about today.
Yeah, good morning and good afternoon, everyone. We hope this is going to be a very informative session for all of you so that we can explain why we're excited about the new advances that have been made at the level of pancreas cancer research and pancreas cancer medicine. If I was to go back 25 years or 28 years when I started working in the field, we used to talk about areas that if we could make progress, it would likely have benefits to patients. Here we are 28 years later, and now we're starting to see some of those original ideas bear fruit. And Andy will be introducing a few topics that my close colleague, Dr. Jaffe, will really regale us with how advances are being made in the clinic, much of it with your support to help all patients with pancreas cancer. And I'll give some comments about some science areas that are now finding their way into the clinic, which I'm very excited about, and I hope you are too. So again, this should be a terrifically informative session for all of you, and we thank Linda and the LustGarten Foundation for organizing it for the good of all patients.
Right. Thanks, Dave. And Liz, how about, love to hear from you and kind of if you share the same level of excitement that Dave has.
Yes, I think Dave said it very well. I'm very excited about where we are today. But just to give you some perspective, my uncle got diagnosed with pancreatic cancer in 1986 and died within 3 months. He asked me what was available. I was an intern training at that time in internal medicine. And even I knew there was nothing available for pancreatic cancer. Today, I can actually say to patients, when we have new therapies being tested for the first time, that this may help you. Imagine that, a new treatment that's just been developed, and it may help a patient today. This is huge. This is so important because not only does it give hope, it's reality. We are seeing responses in our patients, even with metastatic disease. Now, I don't want it to seem like we're helping everyone right now, but we are making really good progress. And why is that? Well, because there's been an investment in pancreatic cancer research over the past 20 years. And I have to say that the LustGarten Foundation has really led that, because when I first started in this field, the NIH was not really prioritizing this disease. They do now to some degree, but again, it can't provide all the funding we need. And what has happened is we've gone from a very small community to a huge community with many different expertise being able to attack the problem. And I have never been this hopeful that I will see patients becoming either cured from early disease or having their metastatic cancers, which are now deadly, turned into chronic diseases that they can live with, like diabetes. I'm extremely excited about our options today.
Thanks. I think you both said it fantastically, and I think really summarized what people are going to hear about in the highlights we talk about today. One, of the importance of research and how the North Star that LustGarten has started with and remains on, and our supporters drive us towards, of, it's going to be understanding the biology that teaches us how to treat the disease. And as Dave pointed out, and Liz, you summarized very nicely, we're now at a point where we're seeing research turning into solutions that are impacting patients today. And one of those, I would start off asking you about a little bit, Liz, is advances that we've seen with chemotherapies. And we saw actually in 2024, the first approval of a new chemotherapy regimen for patients with metastatic disease in more than a decade, as well as a lot more research going on into understanding how best to use chemotherapies in people with advanced disease. So maybe you could start off, Liz, tell us a little bit about what NALIRIFOX is and what this new approval of a new chemo regimen is showing for patients, and then we can talk a little bit more about what to look forward in the future of chemotherapy for patients with pancreatic cancer.
Yes, absolutely. We're very excited that we have now a third possible treatment for first-line metastatic disease. What does this mean to patients? Well, patients, 70% of patients with pancreatic cancer present with metastatic disease. And I think this will change too, and you'll hear from Dave later about some of the work that's ongoing in earlier detection. But right now, we need options for our patients who present with metastatic disease. And so this new therapy is actually not that new in the sense that all of the drugs have been approved for pancreatic cancer, but one of the drugs is given in a slightly different formula. And this formula allows the response to actually be somewhat increased in some patients when compared to the other two chemotherapies that we currently use. Now, in the big picture of things, as I mentioned, 30 years ago, there were no options. Now we have 3 options, but we're still talking about a survival benefit between 7 or 8 months and 12 months. So what this new treatment has done is in some patients, it has increased survival by about 2 months. Now, here's the problem. We can't tell when we determine which treatment to give a patient when they present to us. We can't tell which one will be the best treatment for each patient. As it turns out, some patients will respond to one of those 3 types of chemotherapy. Others will respond to one of the other forms of those 3 types of chemotherapy. So what we set out to do, and this is work that was done not only by Dave Tuvison and myself with the LustGarten Foundation, but with colleagues not only around the country, but also in Canada. We developed a clinical trial where we compared, at that time we had 2 chemotherapies. We had FOLFIRINOX and we had Gem/Abraxane. And we enrolled over 120 patients. We randomized between the 2 chemotherapies, and we took a real look at each patient's pre-treatment biopsies. So what does this mean? We did genetics. We did profiling of the different types of cells within the tumor. We looked at the inflammation. We looked at whether the immune system was seeing these cancers. We want to be able to take a patient's biopsy specimen and be able to say, will they respond to FOLFIRINOX, or will they respond to one of the other 2 chemotherapies that we have available to us? And so we just completed analysis of this study, and we are learning a lot about how to predict who will respond to which chemotherapy. Why is this important? because right now we really have one chance to make a big difference in a patient when they present, right? So we want to give them the best possible treatment we have for them. We want it to be patient geared, right? Specific for that patient. But also chemotherapy is toxic. So we want to make sure that we're not giving them something that's not going to work for that patient. So I think this is a critical study that is going to now lead to new studies where we can not only say to patients, we think you're going to do better with this particular chemotherapy combination, but also we can add additional agents because this study has allowed us to identify agents that might have helped if we had added them onto the standard chemotherapy. So I think we're at a good time. We're going to make a lot of progress with the current therapies we have and hopefully identify new therapies or new combinations that do even better for our patients.
Yeah, no, thanks, Liz. And I'm glad that you brought up the past study because that's really, I think, one of the trials that's going to be a landmark trial in pancreas cancer for a long time and generated an enormous amount of data that we'll keep going back to and learning about more and more about pancreas cancer, what's different, what makes different patients different. As you said, first, to start off, get people on the right chemo at onset, but also to factor in other therapies as they become available. But one of the things that you didn't mention, which I'd love to hear from you a little bit, is it was also a study that was run in kind of a different approach, or approach that people didn't think was really going to be possible in patients with advanced pancreatic cancer, right? Where very data intensive, taking multiple biopsies from patients and being able to generate all of this data. And I think the team really showed that it is possible to do this kind of deep science clinical trials in large numbers of patients with advanced disease. So it'd be great to hear from you a little bit of how the success of not just the outcomes from the trial, but just being able to run the trial is informing how we're thinking about doing clinical trials in the future, especially to approach more of the, personalized or matching the right treatment to the right patient at the right time kind of approach.
Yes, Andy, that's a great question. And I think, we've learned a couple of principles from this study. One, we can do a multi-center study, and this was 6 different sites, and each had a different expertise in trying to understand what's happening in the tumor. So every biopsy done on these patients was actually divided and shared among these 6 sites. Some people had expertise in really doing an amazing job of understanding not just what kind of genetic alterations were in the tumor, but really understanding them at a level that has never been understood before. In particular, mutated KRAS is an example of that. How is that important? Well, this is helping because we now have drugs, and Dave will mention a little bit about this, that can target these mutations, but we need to be able to understand these mutations in each patient. So our data will contribute to making these new drugs even better. So that's one way in which we were able to contribute. A second way is that, I feel very bad and Dave feels very bad, and all of my colleagues around the country, we're humbled by this disease. We're not trying to do this disease on our own. And I feel bad when a patient has to travel all the way to Hopkins because there's nothing for them, in Texas, or there's nothing for them in St. Louis. It's nice to be able to work together as a team and be able to offer new therapies around the country, making it more convenient for patients, and being able, teaching different groups that don't know how to do this for patients with pancreatic cancer. Not every center can. So here we can help centers that haven't been the center for pancreatic cancer actually take very good care of patients with pancreatic cancer without having them travel very far. So I think we've learned a lot about the science, how to do it, how to work together, how we're going to make treatments better in the future because of this study and the future studies that will be able to build on this study, but also making it convenient and helping other centers really take the best care they can of our pancreatic cancer patients.
Thanks, Liz. And I think one of the very important points that you keep bringing up is this is really a gateway to get us to that next generation of therapies that are going to target the real root causes and root drivers of the disease. And this will be a good time to bring Dave into the conversation a little bit. I think if we look at other cancers like lung cancer and breast cancer, where we've seen real significant advances in terms of patient response, it's really been driven by coming up with what we call targeted therapies, or therapies that address the specific mutations, the specific changes in cells that drive them to become cancerous. That's been very tough in pancreatic cancer, right? Partly because we haven't really understood about what makes different tumors different in different patients. We haven't had drugs available that target the key drivers, but that's starting to change. And I think over this past year, we saw an approval of a new drug that targets a very rare fusion in pancreas cancer called NRG1 fusions. But we've also seen huge advances with drugs that target the key driver in most patients with pancreatic cancer. The sort of KRAS inhibitors are called. So I was hoping, Dave, you could talk a little bit about, what it takes to bring these kind of targeted therapies into the clinic, what it's going to take to bring them forward, and what you see as kind of key opportunities in that realm.
Yeah, thank you. Thank you, Andy. And I feel that we are at a watershed moment in pancreas cancer medicine and research, in that there are new approaches available to target the common driver mutations in pancreas cancer. So in addition to having improvements in the systemic therapies that Liz was speaking about, and which the PASS-1 clinical trial that she helped lead with Jennifer Knox have provided to us, we now have, as you've said, targeted therapies for driver mutations in pancreas cancer. And one example is the approval of the fusion oncogene-driven pancreas cancers that have the NRDG fusion, where there are kinase inhibitors that actually inhibit that oncoprotein. And patients with it, they do not have a KRAS mutation. They have this mutation instead. They have a response that could be measured sometimes at the order of months of response to this pill. And this is something we've been looking for a while, a pill that makes your pancreas cancer better. And so it was really amazing. Now, that's not a common form of pancreas cancer. The common one is, as you just said, Andy, with the mutation in a gene called KRAS, spelled KRAS. And most people on the call know about KRAS. This is a gene required for development. We needed it. It drives health during development, but it can become mutated in cancer, and in pancreas cancer, it's the most common mutation we find. And so chemists have devised chemicals which can inhibit this oncoprotein, so the protein form of the gene. And these chemical antagonists look very promising in the laboratory. And more excitingly, they look very promising in the clinic. And so this is a new tool that is not approved, but it looks like it could be a therapy for many of our patients. And so we're all excited about this. We're also, as Liz said, very humbled, because we realize it's going to get our foot in the door for most patients. It's going to enable most patients to see what a response feels like. They will feel better. They will feel healthier. They will be healthier. But it won't be enough to finish. And this is true of targeted therapies for other cancers. Usually the targeted therapy for other cancers that are approved work, but they don't work forever. They're not a durable response. So I think we're going to need to get very astute as to how to treat patients at a personalized level for the other pathways. And this is where the topics that Liz brought up are going to be so essential. Trying to understand what's the next target in a patient that has KRAS inhibitors first is going to be so key. And so, of course, I'm really hopeful that Dr. Jaffe is going to bring the immune system in to be our soldiers to fight the pancreas cancer in patients. And I know she's excited about that possibility too. So stay tuned. And hopefully in the next year or so, she'll tell us if that part works. I'm more on the targeted side of things, chemicals, antibodies, et cetera. And so I think there are going to be combinations that will be effective for patients, as is true of, for example, infectious diseases. But to identify those other targets will require research, will require clinical trials where patients, they sign up for these trials, where we study the patients very carefully, where we get these biopsies that Dr. Jaffe talked about, either biopsies of the blood or biopsies of the tumor. and we study how do they respond to, for example, the RAS drug, and what might be the next drug to give them. There is a field of oncology where this approach has really blossomed, and that's breast cancer, where there's a group of investigators who've developed a method which allows them to predict drugs that might help a patient, probably better than any other field in cancer medicine. And I would love for that approach to be adopted in pancreas cancer. It's harder because sadly the patients are sicker when they're diagnosed as compared to breast cancer, where oftentimes you're diagnosed with a localized tumor and you yourself feel fine. You just have a bump that somebody finds either physically or by scans, et cetera. In pancreas cancer, as Liz described her, her relative, patients present, they don't feel good. And so every time you have a cancer patient that doesn't feel good, it's tough, no matter what type of cancer. Pancreas cancer, sadly, this is one of our challenges. And so, but I do think we're going to devise smart approaches to identify second drugs and third drugs. I use the word drug, generally it could be a biologic, it could be an immunologic, could be even a novel form of radiation. One could even say maybe microsurgery will start to be real. And so we're open-minded as to what that next drug will be, but we want patients to have that chance to enroll on trials where they can fight the disease as the scientists and the clinicians are fighting it alongside them. For that reality to take hold, we need, as Liz said, we need a network across the country, maybe across the globe, where we can support this concept that pancreas cancer patients need special care, because by getting special care, they'll have the most shots on goal, they'll have the most second therapies, and we think the RAS drugs are going to open that door for most patients, and we want to keep that door open and give those second therapies, those third therapies, until there's no more cancer to fight. Liz talked about chronic disease. That'd be great. We want the patient to go back to what they were doing before they had their diagnosis. And we finally, I think, are starting to see this. And I don't think we're going to spend much time on Liz's breakthroughs in immunotherapy, but they're amazing. And they're separate from this whole RAS drug. And so I'm real hopeful that when she brings her special teams in with the RAS drug, maybe something great will happen. I'm trying not to put too much pressure on her, though, and because I need her to be very sober with her experiments and her clinical trials. But we're real excited right now. And RAS is one, the fusion tumors are another. There are going to be other targeted therapies like that we're going to learn about from the genomes and the transcriptomes of the tumor. So getting a good biopsy and/or getting a really good blood test will be essential for every patient, and I hope that becomes standard practice. So,
thanks, Dave. I think we could all agree that the data specifically around these KRAS drugs is very exciting, but also, as you point out, the potential that this is opening up to move forward and how it dovetails very nicely with what Liz just told us about the past study and being able to do these large coordinated trials going forward. And we're getting some questions that are relevant to things that we're talking about. I'll let you answer the one about the I-SPY trial, Liz, quickly, and we'll move on. And we will get to all the questions, but just a reminder to folks, if they do have questions, type them in, and either we'll address them as we discuss, or we'll save some time at the end. But if you...
Right. So the I-SPY trial. Yeah, so that's the name of the trial that Dave had mentioned in breast cancer, and it's been ongoing for quite a while. It's a very interesting trial, but it's not AI driven at this time. So, it's interesting about AI. There's a lot of AI work being done, as it's basically in its earliest stages in healthcare. And one of the principles of AI is that AI utilizes data that's been generated. And so first we have to generate good data that really tells us something about different types of patients and the cancer before we can teach a computer to learn about that data and then predict in other patients. So we're still at the stage of trying to understand what's going on. So like the past study, a lot of that data can go into a computer program to help us learn. And that may be the start of trying to use AI to determine who's going to respond. But the I-SPY study is more of what we call a platform study, where you start out profiling patient's tumor the way we did in PATH. And if you see a genetic alteration or a pathologic alteration, such as HER2/neu expression, right? We have HER2/neu targeted antibodies, you treat with that. And you start to compare patients on this platform study, and you add different treatments and new patients onto the study, so you could start to accumulate a lot of information, again, that can be used in the future to develop AI programming.
Thanks. Thanks, Liz. That's really critical of the importance of the other generating data that's very high quality, very clean and linked with outcomes and moving forward. And I think it points to I-SPY as something that predates the excitement around AI or the real explosion in AI, and still could show that we can make good decisions about how to treat patients. So now as we move in with the actual intelligence of the smart oncologists, as well as the artificial intelligence, we hopefully will be able to do even better and accelerate that even faster. But I want to back up for a moment to another point that Dave made before, Liz, which is that often patients, when they show up to the clinic, they're already very sick. And one of the things that's an additional challenge with pancreatic tumors is that they make patients very sick. One of those things is causing a process that we call cachexia, which is the severe weight loss and muscle wasting that goes along with the disease. And that's often limiting and frustrating for you and others of what you can do, trials patients can go on, or how aggressively you can treat the cancer. But there was a bit of a shift and a breakthrough, and obviously, and some hint that we might be able to do something to make patients feel better and to stop or potentially even reverse cachexia over the last year. Can you talk a little bit about this exciting work around GDF15 and cachexia?
Yes, absolutely. And we are very excited about this because, as you've mentioned, Andy, cachexia really affects the quality of life and can cause early death. And certainly it makes it very difficult for us to treat patients because they're just not in very good shape to handle the chemotherapy or other treatments that we have. So there's an experimental drug that was tested in an early phase trial in patients called ponsegramab. It's an antibody that inhibits a pathway called GDP15. And this is an important pathway because what we're starting to learn about this pathway is that it signals to the brain, actually. And it's the brain that's signaling to the muscles and to even fat cells, your fat reserve, to actually organize and start to decompose. So by inhibiting this pathway, the concept is that you could prevent that signal, and then patients' muscles would stay intact. They won't lose so much muscle weight, they won't lose so much fat reserve, and they'll remain more healthy. They'll be able to eat and gain weight. So this was tested in a nice size patient population. And on average, there was a 5% increase in weight gain, probably around 5 to 6 pounds in just 6 weeks on treatment. And that may not sound significant, but in a patient with cachexia, not only are you putting weight on, it means you're able to eat again. So you're able to utilize the food that you're eating, eat good food, not feel sick, and be able to start to put the weight on. So this is very important because simultaneously with this study coming out, my team, led by a young investigator, Dan Zabransky, and mentored by myself and an aging expert named Ashani Warantada, we mentored Dan on looking at pancreatic cancer and comparing differences in patients who are over 50 versus under 50, and were there any signals within particularly their fibroblasts, the stromal cells. So what are fibroblasts? They're basically, we call them stromal cells, but they're cells that are normal cells in any organ that typically supports normal cell growth, normal cell function. But unfortunately, cancer cells co-opt them and take them into their domain and ask the fibroblasts to help them grow. Well, what we found was that GGF15 actually is produced by aging normal fibroblasts, and in animal models enhances tumor growth. And using an antibody that can inhibit it or knocking it out genetically we can prevent or slow that tumor growth. So this pathway may have two roles. It may help with cachexia, and it may also help with inhibiting tumor growth. So I think the next steps will be, can we utilize this pathway and combine it with chemotherapy or other treatments to reduce the cachexia so patients can tolerate the treatment better, but also to enhance the potency of our chemotherapy agents.
Yeah, no, thanks, Liz. It's really striking the results that came out in the cachexia study, not only that it slowed or stopped the weight loss, but that there seemed to be weight gain and reversal. And getting back to the point of really the ability to bring together all of these different complex and interacting systems to understand the impact that the tumor is having locally in the environment on the fibroblasts and how the fibroblasts are supporting the tumor, the impact on the central nervous system and the brain and controlling diet and weight loss. And it's not something that, we would understand if we didn't have this deep look at all of the different organs that are involved and all of the different interactions. And I think really points to some excitement of having therapies that can both target the tumor and target the health of the patient overall is very exciting. And I think some of this points back to and a question that had come up in the chat around, well, what are the signs? What are the early events that happen as somebody is developing a pancreas tumor, and how could you think about that? And can we do better at identifying people at those early stages of pancreas cancer and help to move them forward, move forward the diagnosis and find them earlier? And I think we've seen a lot of advances over this past year, particularly with AI tools becoming available and accessible, and people being able to look at deep data sets, whether it be imaging data, whether it be clinical records, whether it be using AI to apply to research data to discover new things. We're learning a lot more about what are the signs of early cancer and how does early cancer develop. And I know, Dave, this has been a real passion of yours, of that not only are we not doing well of finding pancreas cancers early, but we really need to move on and think about this in a new way and a different way. And I think that tide is starting to turn. So I wonder if you could comment a little bit on, what you've seen coming out in the field of early detection. Are we moving into a new era where we could think about this differently, and kind of where do you see that going overall?
Yeah, I mean, thank you for bringing up this topic, Andy. The early detection of pancreas cancer would likely lead to much better outcomes for patients. And we can say that with some degree of confidence because that's the case for other types of cancer. And we do have some evidence from a study that has been going on at Johns Hopkins for several decades now, where they have carefully been studying families who are at risk for pancreas cancer. They have several relatives with pancreas cancer, and the geneticists have identified in these families, many of them, but not all, a potential gene that predisposes those families to the cancer. But either way, these individuals are assessed multiple times a year by visiting the doctors, getting blood tests, and getting imaging tests, radiologic imaging, either using ultrasound or other anatomic imaging methods such as CT scans or more commonly MRI. Now, a study that was published just a few years ago by this group, led by Michael Goggins, Alison Klein, and Mimi Canto, and inspired by the early work of Ralph Ruban in creating the Familial Pancreas Cancer Registry, they published a few years ago that in their large cohort of individuals who are not yet patients, coming from families at high risk, they, for the first time, were identifying patients with tiny pancreas tumors. And these tiny pancreas tumors that they identified in patients, when the patients went to surgery, oftentimes were even confined to the pancreas, meaning not spread or metastasized outside the pancreas. These individuals showed an increased survival from the time they were diagnosed, when they were studied then for over a decade. This is really the first evidence, in my mind, that early detection could have a profound influence on how patients do. And I feel that when you that should be again an exemplar for all of us. That could we devise early detection strategies that work for patients at high risk, such as this group, but also the general population, which is much larger, 80%, 80% plus are in that category. We now know that about 1 out of 60 to 1 out of 70 people in America, United States of America, develop pancreas cancer. We also realize that most of those patients will lose their battle to pancreas cancer. Well, another study from Johns Hopkins showed that if you were to look at the pancreas of patients who don't have pancreas cancer, and this is a study led by Laura Wood at Johns Hopkins and her colleagues, most of us have the signs of a precancer in our pancreas. And the precancer is called pannin, like panini with the last I out. So pannin, very medical term that Dr. Ruban invented that we all adopt. We all have early stage pannin, but we don't all have what we call advanced pannin, or the last stage of pannin before invasion. This is a very important point because lots of us are interested in developing tests for early pancreas cancer. What we need is a test not of early pannin, but of intermediate or advanced pannin. And then we could say, well, what's that test going to be? Andy brought up the concept of having computational support, machine learning, natural language processing, artificial intelligence, use the term that pleases you. Yeah, those methods work when there's data. So what's the data going to be? Well, we have blood tests. Blood tests give us things circulating in the blood. It could be signs of early pancreas cancer, be it the gene we talked about, the KRAS mutation, be it other mutations. We also have proteins made by the tumor. There's a biomarker most of you have heard of called CA19-9. This is an aberrant glycosylation which occurs in most patients with pancreas cancer. And there are other things like that. There are other circulating entities that come from the cancer or the stroma. Liz spoke about how the fibroblasts were different in the GDF15 treated patients. That's totally fascinating because the major cell type in the pancreas tumor are the fibroblasts. So one would think the fibroblasts would have their chorus of disease in the blood. So we need to figure out what all that is. What are the songs of the cancer we can see in the blood? Terrific radiologists are trying to develop better ways to do imaging. We have a big approach at Johns Hopkins working with the group there to develop fancier ways to see tumors before the radiologist's eyes see the tumor. And so that's anatomic, so the shape, for example, of the tumor. But combining that with molecular details creates a field called radiomics, where it's radiology plus some version of omics, which are measurements of biological processes. That is computational science. That is artificial intelligence, if you'd like to use that term. We need people who think like that, who are excited by that. Unfortunately, I'm too old to get excited. I'm excited, but I'm not going to be a player. I can just support people to bring them into the field. I think the same is maybe true of Liz. We're here as cheerleaders to get new smart people into the field to take this on as their approach that they can move forward. So I'm hopeful that'll work. What I'm also hopeful is that Liz can bring the immune system in to be our detector. Now, she's a world-class immunologist. She's the leading cancer immunologist for pancreas cancer, without a question. The immune system knows we have cancer every time, but unfortunately, due to what the cancer does, it prevents the immune system from fighting the cancer in different ways. But it knows it's there. And if there was a way to make an immune detection signature of pancreas cancer, that would be cool. And I know Liz thinks that would be exciting. We don't have it. It's like I'm telling you about the stuff that doesn't exist, but we're trying to encourage people to work on all those topics. And Andy and Liz and I just sponsored a think tank just a few weeks ago because the Lustgarten Foundation is very passionate about this topic. It doesn't mean we have it solved. We need your help. to tell us how interested we should be in this topic. I think we should push hard in early detection. But we brought in smarter people who work on brain cancer and ovarian cancer and other diseases where they have the same problem. Patients are not picked up early. And the scientists and doctors were from Europe, the US, other countries, and we sat around tables and were really critical with each other about what we know and what we need to know. And we came up with some really great ideas at this meeting. And we would like to, make a big push in this area to bring in computational scientists, biologic scientists, clinicians, engineers, people with new ideas, so we can critically test these ideas and support, creative new insights. If we can detect the cancer early, we will help so many more people. There's no doubt in any of our minds. And then the clinical trials that Liz is leading on, we'll have, people who will qualify for the trials. We will learn what's the best medicines. Then we can apply that knowledge to most patients who are diagnosed a little bit later. So this is a big deal for the Lustgarten Foundation, a big deal for all of you out there. And I'm quite hopeful that we're going to see a sea change in this space. But I would like, Liz can, really improve upon any of the comments I made on this topic, and maybe some of you will work on it.
No, thanks, Dave. And I agree with you. I think the workshop that we just had a couple of weeks ago was very exciting. People are ready to tackle this problem in new and inventive ways. And it's really, I think, the role of Lustgarten Foundation and similar organizations to support that level of innovation and really challenge people to think about things differently and to take risks, because we know that the patients and the families can't wait, that these diagnoses are coming and we need to do better today. And just as a reminder to folks also, so this on today's webinar, we get to give a little bit of a smattering of a lot of different topics. We do have webinars coming up in the future where we'll dig in on some of these topics more in detail. Early detection, I imagine, will be one of those over the next year, as that's a very exciting area. We also talked a lot about immunotherapy a couple of months ago with Bob van der Heide, Dr. Bob van der Heide from University of Pennsylvania, who's an expert in immunotherapies, works very closely with Liz. But I see a lot of questions coming in about vaccines and immunotherapy. So Liz, if you don't mind, we'll put you on the spot a little bit. If you want to make a couple of points. I know this is the area you more than anyone has pushed the pancreas field to really think about the immune system, and it's an exciting area. If you want to comment on, why vaccines are so attractive, what's happening there, and immunotherapies more generally, how they fit in with everything that we've talked about so far.
Yes, absolutely. So we've been looking at vaccines for quite a while in this disease, over 30 years. And what we had seen when we tested our vaccines in patients is that the vaccine will activate the soldiers, as Dave said. I like that word too. The cells that are going to actually seek and hopefully destroy the tumor. And we could see that in the blood. The problem is that we've also learned that pancreatic cancer is quite complex. And so for these vaccine-induced armies to get into the tumor, it takes a lot of work. A lot of signals have to be happening that allow them to come in instead of inhibiting them from getting in. And that stroma, the fibroblasts, are a group of cells, multiple cell types. And Dave actually identified some of the critical cell types within these, we call them cancer-associated fibroblasts. But they provide a lot of signals between the tumor and the immune cells to inhibit them naturally from getting in. And it makes sense because it's a natural response in any organ to prevent autoimmunity. And that's the signals they're sort of co-opting. Well, since the development of these other agents called immune checkpoints, and you've heard of Keytruda, Optiva, some of those important drugs that target anti-PD-1. Well, there's a whole panel of these now. We've learned a lot about how to activate and downregulate these army of T cells, army of immune cells. And so by giving these cells, we can actually, by giving these antibodies to these signals, these immune checkpoint antibodies, we can actually facilitate these T cells from getting from the blood, where they're naturally circulating after a vaccine, into the tumor to fight the cancer. And so one of the studies we've been doing, and Lustgarten has certainly supported a fair bit of this work over the years, is we developed, like the I-SPY, a platform study where we started out with vaccine and then started adding some of these other immune checkpoints. And we're now at a point where, at least in patients who are surgical candidates, we can actually give our vaccine plus some of these immune checkpoints ahead of surgery and actually see that the tumors have responded and are actually becoming necrotic. And so we did this initially, and now we have a bigger study that's about to start enrolling to see if this is something we can take to FDA approval. So how do we think about our vaccines now? Well, we think about targeting KRAS, right? So you've heard a lot about it from Dave. Well, it turns out that mutated KRAS is a very important genetic alteration in pancreatic cancer. In fact, 90% or greater pancreatic cancers express one of 6 or 7 forms of this mutated KRAS. In addition, we know that the earliest precancer, the pannins, will express it too. The cancers can't survive without mutated KRAS, and that's important because there are different kinds of vaccines, and targeting mutated KRAS makes a lot of sense in this cancer, not only for treatment, if we can give it with these immune checkpoints, because the cancer is so dependent on this mutation. But also, we've now started to look at it in the setting of intervention. So if a patient has a high-risk lesion like a pannin, the FDA has allowed us to start to test this vaccine in patients or in healthy people who are at high risk to try to prevent the cancer from forming. So that's one example. You've certainly heard about Alicia's vaccine, and that's another KRAS targeted vaccine, and they're in a large multi-center study to see if it really will have benefit after surgery, after chemotherapy, to prevent the cancer from coming back. And then the third form of vaccine that you've probably heard about are these mRNA vaccines, again, similar to the COVID vaccine, where we biopsy a patient's tumor. We try to figure out what are the mutations other than KRAS that they express and try to activate the immune system against those mutations. And in a small study out of Memorial Sloan Kettering, and again, supported in part by the Lustgarten Foundation, that study too has shown some evidence of preventing patients after surgery, after chemotherapy, from having their cancer come back. But these are all small studies. We're talking about 10, 20 patients at most. So really, we have to see which of these vaccines, and I suspect they're all going to have a role. But the important point is to make sure you give the right immune modulators, these immune checkpoint agents, to make sure whatever T cell you can activate can actually get into the tumor and do its job to kill the tumor. So I think, stay tuned. A lot of excitement, as Dave has said. I'm excited. For the first time, we're really seeing patients respond. We're even seeing patients in metastatic disease respond to our vaccine plus immune checkpoint therapy. But again, these are small studies, so we still have to learn more of how to improve on them and make them work for more patients with this horrible cancer.
Thank you for summarizing that work so clearly and concisely. And I think it is what I think is particularly exciting there is, one, on the vaccine side, we're seeing responses with vaccines generated in multiple different ways. Peptide vaccines like you're doing, some peptide vaccines with some special modifications like Alicia is doing, the mRNA vaccines. People generally are seeing at least the trend that these can work, but it's finding the right time to give them in terms of stage of disease and the right combination of treatment to give them with. And I think that's a lot still that we're figuring out of how early can you treat somebody at risk? Hopefully with these vaccines that are very safe, we can go fairly early. And as it gets later, what other drugs do we want to combine them with to have maximal effect? And I think that's where your platform study and patients going for surgery has been really informative to be able to look at the tissue and see how the immune system is performing there and knowing what to add in. So we're getting close to time here, and I know there's a few questions we didn't get to. We'll try to follow up with people offline if we can. But I do want to give both you and Dave and Liz both a chance to kind of close us out here and give a little bit of a big picture sense, like how we started, about, where you really see work going, what you're excited about, and what our community should be able to look forward to this time next year when we look back again and think about where successes have been. So I don't know, I'll let you go first, Dave, and lead us off.
I'm hoping by this time next year that we'll have, a clearer indication as to whether this new type of targeted therapy is effective in a randomized clinical trial. There's a phase 3 trial ongoing and enrolling. And I think the interim look at the data should be available within a year, given that the popularity of that trial. If that is the case, then that would be the first example. I suspect, as Liz said, there will be many such examples of RAS therapies because there are multiple approaches out there, which is great, for the field. But that will herald then a moment for the field and for patients. Because if it does get approved by the FDA, then it becomes, as we talked about in the beginning, an approved therapy. Then it's a question of when to use it. The trial ongoing right now is to use it in patients who've already had first-line therapy. But Liz and I and many other investigators, our feeling is, what if we used it first? And, we could debulk, meaning shrink the tumor in a way that is not as damaging to the rest of the patient's body. I think a trial that Liz and her colleagues will be excited to do would be to combine it with the GDF15 antibody. And who knows, that I'm not making stuff up. That actually, things like that could really change the field. And so, Andy, a year from now, my hope is we've gone from small patient studies, which Liz very carefully talked about. We get very excited by tiny numbers of patients and then disappointed when the population gets bigger. So we have to be sober to that reality. And I think small randomized trials, which is what the PAS-1 clinical trial was, it was medium-sized, taught us a lot. We need that. We need that kind of structure where it's like a medium-sized randomized trial so that we are enthusiastic in a rigorous way. And if we can get a few of therapies, elevated like these, then I think coming in with Liz's various immune therapy approaches, different types of cell therapies, vaccine therapies, cytokine therapies, all of these things that Liz is a master at, I think we're going to start to see some amazing opportunities for our patients that many of you are supporting who are on this webinar. So again, I'm putting the pressure back on Liz, but I guess in the laboratory, yeah, we have things to learn. We need to learn why. We need to learn why some patients respond and why others don't. So the job of the scientists is to get the samples and work really, really hard to try to understand this and be open-minded as to what are those other pathways that we need to bring into this. And so the last word should come from our master clinician.
And I'll give it to Liz in a second, Dave, but I think you summarized nicely, like the three legs of the stools are coming together, right? The scientists are there with the great ideas to test. The clinicians have figured out the right ways to test them and all the things to measure in patients. And now having drugs available that can really make a significant impact in the disease is where we're really going to start to see an acceleration. And I think that's a great time to give it to our chief medical advisor and one who's been guiding us on our clinical strategy for so long to tell us why we should all be excited.
Yeah, I couldn't say it better than Dave and Andy, but I do want to say that there is a fourth leg to that stool, and it's everyone on this webinar and our patients. It's really a team effort, and the LustGarten Foundation, our other supporters, all our patients, and all the patient supporters, their family and other friends. This really, the only way we're going to make a big difference is if everyone comes together and works together. And the fact that our patients, are willing to go on our clinical trials. I mean, we're very thankful because we could not learn without all of our patients who are willing to test new drugs with the hope that it's going to benefit them. And as Dave said, I think more than ever, I am very hopeful that we are benefiting our patients now and will continue to do even better over the next couple of years.
Well, Dave, Liz, thank you for sharing. Always enlightening, always challenging, always energizing. to speak with you and to learn about how much potential there is to move forward. So with that, I don't know, Linda, if you want to come back to help me thank Dave and Liz for their time today and help to close us out on this webinar on this great message of hope and dedication to our patients and our families.
Yes, thank you all so much. This was really an illuminating conversation, and I know that our community really looks forward to hearing from all of you and from hearing about the work that they are supporting. We could not do this work without all of our supporters, and we certainly could not do our work without our guides and our leaders, Andy, Dave, and Liz. So we so appreciate everything that you do for the foundation, and we look forward to a terrific year in research and a terrific year in discoveries. Our next LustgartenLIVE will be on March 7th. It actually will be International Women's Day, and we are very excited that we will be hosting this live symposium at Johns Hopkins under Dr. Jaffe's leadership. And we have terrific speakers, and we'll be really focusing on how women leaders are transforming the field of cancer in general, and then, of course, in pancreatic cancer research. So we look forward to all of you joining us on March 7th and to other LustgartenLIVEs throughout the year. So thank you all very much for joining us. And again, Dave and Liz, thank you for giving up your very precious time to our community. We really appreciate you. And to our audience, again, many thanks for all that you do for the Lustgarten Foundation and for your support. So thanks, everybody.
Thank you all. And we'll respond to the questions that we didn't have time to address during the live webinar.
Great. Thanks, Dave.